So, forgive the dodgy editing. What we're going to think about next then is how DNA actually carries out its function. So we've discussed previously that DNA controls a cell's activities because it acts as a genetic code for protein synthesis. But what we want to think about now is how does that genetic code actually result in proteins being synthesized. So if we consider and look at an example of a DNA molecule, as we've already discussed, we know that a DNA molecule is made up from two polynucleotide strands joined together via complementary bases. And the key to the genetic code are the bases, and specifically it's the sequence of those bases. So if I just give a sequence of bases as for example, so there we've got a sequence of bases running along the DNA molecule. The way in which the genetic code works is that every 3 bases is going to actually determine and code for a specific amino acid. So these 3 and these 3 and so on and so forth. So the DNA base sequence of TAC is going to determine and code for a particular amino acid that you find in the primary structure of a polypeptide chain. So the 3-base sequence of TAC will determine the fact that the amino acid methionine, and if you remember when we discussed the structure of proteins, we can shorten the name of the amino acid to the first 3 letters. So we have methionine, CCC, 3 cytosines will code for the amino acid glycine. TAC is going to code for methionine again. And just to finish off my example, a GG is going to code for the amino acid serine. So the sequence of these 3 bases determines a particular amino acid, and each sequence of 3 bases, which we are going to refer to as a triplet code. So each one of these is a triplet code. Each triplet code determines a particular amino acid, and so the sequence of the triplet codes therefore determines the sequence of the amino acids within a polypeptide chain. So each of these amino acids are joined via peptide bonds. We know then that the primary structure, the sequence of amino acids within the polypeptide chain, ultimately is going to determine the overall structure of the protein. And without revising the whole of protein structure, remember you can have fibrous proteins, you can have globular proteins. Proteins can also be quaternary, so they might be made up from just one polypeptide chain. They might be made up from two or more polypeptide chains. But if I give an example there of a globular protein with a tertiary structure, it relies heavily on its three-dimensional shape in order to carry out its function. We know that the sequence of the amino acids determines where bonds and large folds take place, which actually maintains the tertiary structure of the protein. And let's just say, for example, that this particular protein is an enzyme. And finally then, the enzyme has the ability to catalyse all of the cellular reactions, which ultimately therefore gives the cell characteristics. So, for example, this might be a muscle cell. So the way in which DNA acts as a genetic code is that it's the sequence of the DNA bases, which appear in sequences of threes, so a triplet code determines a specific amino acid, which then is found in a specific sequence within the polypeptide chain, which then determines protein structure, and proteins such as enzymes are going to catalyse all of the cellular reactions to give a specific characteristic of the cell.